Hi, good morning, everyone. In last class, I have covered some basic information about neurotransmitters, like definition of neurotransmitters and classification of neurotransmitters. So there is a function classification and chemical classification and location of the neurotransmitters and functions of neurotransmitters. So in today's class, uh, I'm going to explain about neurohumoral transmission. Okay, so what is neurohumoral transmission? First, what is the definition of neurohumoral transmission? So, neurohumoral transmission is also known as neurotransmission, and it is also known as synaptic transmission. Okay, so what is the exact definition is a process of transmission of nerve impulses. Okay, the process of transmission of nerve impulses from one neuron to another neuron or neuron to effector organs by the release of neurohumoral substance that is known as neurohumoral transmission. Okay, got it? Definition: the process of transmission of nerve impulses. So the process of transmission of nerve impulses from one neuron to another neuron or neuron to effector organ by the release of neurohumoral substance. Okay, so that process is known as neurohumoral transmission. Okay, so what are the steps? The neurohumoral transmission process consists of 5 steps. Okay, so that is impulse conduction, transmitter release, and transmitter action on post-junctional membrane, and post-junctional activity, and termination of the transmitter action. Okay, after showing its action, there is a termination of neurotransmitters. Okay, and coming to the first step, that is impulse conduction. Okay, so in this impulse conduction, so normally in neuron, the resting membrane potential is minus 70 millivolts. Okay, in neurons, the resting membrane potential is minus 70 millivolts. So first of all, what is membrane potential? Okay, so can you define membrane potential? So the membrane potential is the differences between the differences in electrical current. Okay, the differences in electrical current in between interior and exterior of a biological cell, that is called as membrane potential. Okay, so the neuron resting membrane potential is minus 70 millivolt. Okay, so what is membrane potential? Membrane potential is the differences in electrical current. Okay, the differences in electrical potential, nothing but the differences in electrical potential in between interior and exterior of a biological cell. Okay, so interior is nothing but inside of the cell, exterior is nothing but outside of the cell. So there is a differences in electrical potential in between inside and outside of the cell. So that is called as membrane potential. Okay, so here the resting membrane potential is minus, okay, minus 70 millivolts. Okay, the millivolt is nothing but is a unit of voltage. So the minus 70 millivolts, the resting membrane potential. But when the neuron is exposed to stimulus, okay, so when the stimulus is applied on the resting neuron, okay, then what will happen in that resting membrane potential? So after exposure of stimulus to the neuron, the resting neuron, in that cases, the resting membrane potential is minus 70 millivolts. So then immediately what will happen in this stage? There is an increase in the opening of sodium channel. Is nothing but there is an activation of the sodium channel. Okay, so then after the activation of sodium channels, nothing but there is an opening of sodium channel. So then what will happen? There is an influx of, okay, there is an influx of sodium ions into the neuron cells. Okay, so then immediately what will happen? There is a changes of membrane potential. Okay, minus 70 millivolts to minus 55 millivolts. Okay, initially the minus 70 millivolts, but when the stimulus is applied to the resting neuron, then what will happen? There is a membrane potential is increases minus 70 to minus 55 millivolts. Then what will happen in that cases? Then again further opening of the sodium channel, further activation of the sodium channel, and more opening of sodium channel. So more and more opening of sodium channels in that cell, then what will happen? There is a high influx of sodium ions into this cell. Okay, so nothing but the positivity charge inside the cell is increases. Okay, the sodium ions, the positive charge, the sodium ions is increases in inside of the cell. Then immediately what will happen? The membrane potential is will reach from minus 55 millivolts to plus 30 or 40 millivolts. Okay, normally the resting membrane potential is minus 70 millivolts, but when the stimulus is applied to the neuron, then initially opening of sodium channels. Okay, then slightly the membrane potential will changes from minus 70 millivolts to minus 55 millivolts. Then further opening of more sodium channels. Okay, so further opening of more sodium channels is nothing but the high influx of sodium ions. So high influx of sodium ions into the cell. So there is a more sodium ion concentration in the cell is increases, means nothing but the sodium ion concentration is increases in inside of the cell. Then the membrane potential will increase from minus 55 millivolts to plus 40 or 30 millivolts. Okay, so total that process is known as depolarization. Okay, so that is known as depolarization. Okay, then what will happen after depolarization? Okay, then the repolarization is also occurs. Okay, so the repolarization is occurs due to two reasons. So there is an inactivation of sodium channel and activation of potassium channel. So as a result of activation of potassium channels, then what will happen? There is a efflux. Okay, flux is nothing but the movement of ions from inside of the cell to outside of the cell. Okay, so nothing but the potassium ions are out from the cells. Okay, so slowly the repolarization is occurs due to activation of the potassium channel. In depolarization state, there is an inactivation of the potassium channel. In repolarization stage, there is a inactivation of sodium channel and activation of potassium channels. So then after repolarization, then slowly the next phase is hyperpolarization. So the hyperpolarization is occurs due to what? The hyperpolarization is occurs due to there is a high efflux of potassium ions or closing of the potassium channels. So high efflux of potassium ions means there is a the potassium ions, there is a movement of potassium ions from inside of the cell to outside of the cell. So then after hyperpolarization, okay, so after hyperpolarization is nothing but the lower membrane potential. Okay, the resting membrane potential is minus 70 millivolts. So hyperpolarization is nothing but below that resting membrane potential, the lower membrane potential. So the hyperpolarization is occurs due to more efflux of potassium ions. Okay, and then hyperpolarization phases will becomes to resting state. Okay, so the lower membrane potential again, the slowly it will becomes to the resting membrane potential. Okay, so that is known as resting state. So the total consists of what? Stimulus. Okay, when the stimulus is applied to the resting membrane potential neuron. Okay, so the resting neuron, the resting neuron, the membrane potential is 70 millivolts. But when the stimulus is applied to that particular neuron, then what will happen? The voltage will be increases minus 70 millivolts to minus 55 millivolts. Then there is an activation of sodium ions. There is a depolarization process happens. So the high influx of sodium ions here because of more and more opening of sodium channels. So then what will happen? There is a high influx of sodium ions into the neuron cells. Then we will get action potential. Okay, so as a result of the depolarization, then here we will get action potential. This is a peak effect. Okay, this one is action potential. So exactly what is action potential? Okay, when neuron is resting condition, in that cases, when the stimulus is applied that resting neuron, okay, then immediately the resting membrane potential of the sorry in resting membrane potential of neuron is minus 70 millivolts. Okay, so but when the stimulus is applied to that particular neuron, then what will happen in that voltage, there is a voltage is increases. Okay, minus 70 millivolts to plus 30 or plus 40 millivolts. Okay, total that process is known as depolarization. Then we will get action potential. Okay, so after action potential, then what will happen? Transmitter release, second step in neurohumoral transmission. So this one is total action potential phases. Okay, so action potential phases is stimulus. Okay, application of stimulus. Second one is depolarization. Third one is repolarization, hyperpolarization, and resting state. Okay, after we will get action potential, then repolarization processes happen, then hyperpolarization, then resting state. So in repolarization process, the potassium ions are activated, there is a efflux of potassium ions, then it may lead to hyperpolarization. Then slowly the cell will becomes to resting membrane potential. Okay, so here after we will get action potential. Okay, so when the stimulus is applied to neuron, because the stimulus is received by dendrites. Okay, so then what will happen? The membrane potential is increases. Okay, so in normal initial phase, membrane potential is 70 millivolts, but when stimulus is applied to that neuron, then the resting membrane potential is increases from minus 70 millivolts to plus 30 or plus 40 millivolts. Okay, then we will get action potential here. Okay, then that action potential. Okay, so that action potential is when arrived at the nerve terminal. Okay, so in neurohumoral transmission, the first step is impulse conduction. How the nerve impulses, it will be conducted along the neuron. Okay, so how it will be conducted when the stimulus is applied to that neuron. Okay, then we will get action potential because of the depolarization process, because of the high influx of sodium ions into the cell. Okay, so after arrival of action potential, okay, action potential is reaches to the presynaptic nerve terminal. Okay, so this one is presynaptic nerve terminal and this one is postsynaptic nerve terminal. So so when action potential is reaches to the presynaptic nerve terminal, then what will happen? See, this arrow mark is action potential. So what is action potential? So this one is action potential. Okay, so second phase. What is second phase in second step? Transmitter release. Second step is depolarization of presynaptic nerve terminal. Here, so the depolarization of the presynaptic nerve terminal. Okay, so here then which ion channels are activated? That is calcium channels are activated. Okay, so in the production of the action potential, here it is the sodium channels are activated. Here during the release of neurotransmitter, so which ion channels are activated? That is calcium channels. Okay, so the depolarization of the presynaptic nerve terminal. Okay, because of the activation of the calcium channel, so there is a high influx of calcium ions into the cell. Okay, which cell? That is presynaptic nerve terminal. This one is presynaptic nerve terminal. Okay, then afterwards third step is the calcium ions are triggers the release of neurotransmitters from vesicles. Okay, so here after second phase, there is a high concentration of calcium ions is available in presynaptic nerve terminal. Then what will happen? That calcium ions are stimulate the release of neurotransmitters from vesicles. So what will happen here? So these are the synaptic vesicles. Okay, so these synaptic vesicles are fuses with presynaptic nerve terminal membrane. So this one is presynaptic nerve terminal membrane. So the synaptic vesicle is fuses with presynaptic nerve terminal. Okay, then what will happen? There is a release of neurotransmitters. Okay, so there is a release of neurotransmitter into synaptic left region. Okay, So this one is synaptic left. What is synaptic left? The space between presynaptic neuron and postsynaptic neuron. Okay. So then the neurotransmitter is released into synaptic left. Then afterwards, what will happen? That what the neurotransmitters is binds with the postsynaptic neuron. So this one is postsynaptic neuron. So within the postsynaptic neuron consists of receptors is there. Okay. So these are receptors. Okay. This one is receptors. So this neurotransmitter is binds with receptors. Okay. Then after what will happen? After binding with the receptors, then opening and closing of channels. Okay. So this receptor consists of channels. So, okay. So after binding of neurotransmitter with the receptors, then opening and closing of these channels, then what will happen? There is a changes in the postsynaptic membrane potential. Okay. The membrane potential in postsynaptic neuron is changes. Okay. So there is a changes in membrane potential in postsynaptic neuron. Then what will happen? That action potential. Okay. So this action potential, either this there is a changes in postsynaptic membrane potential. So that action potential, okay, it is propagated through next cell. Okay. Then this one is presynaptic neuron, this one is postsynaptic neuron. After arrival of the action potential, there is a release of neurotransmitter. Then neurotransmitter is binds with what receptor? Those receptors is present on the postsynaptic neuron. Okay. Then what will happen? There is a ion channels. Okay. There is a ion channels which are present in receptors. Okay. So depending upon the opening of that ion channel, okay, depending upon the type of that ion channel, there is a influx of positive ions, or there is an influx of negative ions. Okay. So depending upon the type of the opening of the ion channel. Okay. So then the membrane potential in postsynaptic neuron is somewhat changes is occurs. So that kind of action potential is propagated to next neuron. And after that, the neurotransmitter is inactivated or transported back into the presynaptic now terminal. Okay. So this one is process of transmitter release. Got it? First thing is impulse conduction. Okay. So how action potential is produced. Okay. When the normal neuron, okay, in resting condition, in a resting condition, the neuron is exposed to stimulus, then what will happen? There is a production of action potential. The action potential is occurs due to opening of the sodium channel. So there is a high influx of sodium ions. Okay. Normally the membrane potential is minus 70 millivolts. But after getting of the sodium ions into the cell, the membrane potential is increases from minus 70 millivolts to plus 30 or 40 millivolts. Okay. That potential, that action potential, it will arrive at the presynaptic now terminal. Then what will happen? Activation of calcium channel, then release of calcium ions into the cell. Then calcium ion concentration is increases in the presynaptic now terminal. Then what? The synaptic vesicle is fuses with presynaptic now terminal. Then what will happen? There is a release of the neurotransmitter into synaptic left. Then it is binds with receptors, the receptors which are present on the postsynaptic neuron. Then afterwards, what will happen? There is an opening and closing of channels, the channels which are present in receptor. So depending upon the kind of channel opening or closing, so there is the action potential changes is occurs in postsynaptic neuron. Okay, depending upon the type of channel opening and closing in receptor. Okay, so then afterwards, after action potential, then what will happen? The action potential is propagated through another next cell. Okay. So then after that, the neurotransmitter is inactivated and transferred back into the presynaptic now terminal. Okay. So this one is second step. And third step is transmitter action on post-junctional membrane. Okay. So this one is release. After release is binds with postsynaptic neuron. Okay. So the transmitter action on postsynaptic neuron or post-junctional membrane. And third step is post-junctional activity. Okay, in this diagram, these two steps it will be covered. Okay, that is transmitter action post-junctional membrane or postsynaptic membrane, and third step is post-junctional or postsynaptic post-junctional activity. Okay, so presynaptic activity, sorry, postsynaptic activity. So first thing is impulse conduction. Okay, in neurohumoral transmission, the first thing is impulse conduction. Okay, means production of action potential. Okay, so there is after getting the action potential, that action potential it will arrive at the synaptic nerve terminal. After arriving of that action potential at this synaptic nerve terminal, then the second step is the release of transmitter. So after release of this transmitter, then what will happen? Third step. Okay, there is a release of neurotransmitter, then it is binds with receptor. Okay, the receptors is present on postsynaptic neuron. Then what will happen? Okay, depending upon the nature of its neurotransmitter, there is a binding with their respective ion channels. Then here, for example, this one is excitatory neurons, excitatory neurotransmitter. So this red color round is excitatory neurotransmitter. So this excitatory neurotransmitter is binds with the postsynaptic membrane. The postsynaptic membrane consists of receptors. So after binding with the respective receptors, then what will happen? It allow the permeability of cations. Okay, it allow the permeability of what? Which ions? Cations. So that is sodium ions. Okay, so there is a high influx of sodium ions into the cell. Okay, so then what will happen? It may lead to what? Depolarization. So that is excitatory post-synaptic potential. EPSP means excitatory post-synaptic potential. Okay, so the depolarization process is occurs due to high influx of sodium ions, because the permeability nature of this cations is increases. Okay, so high influx of sodium ions and efflux of potassium ions, means there is a movement of potassium ions from inside of the cell to outside of the cell. But here the EPSP, excitatory postsynaptic potential, requires positive charge, that especially sodium ions. Okay, so the sodium ions, there is a movement of sodium ions into the cell from outside of the cell to inside of the cell. Okay, so here normally in resting condition, okay, in resting of the resting membrane potential, in that cases the potassium ion concentration is very high in that inside of the neuron. Okay, so here in depolarization process or excitatory postsynaptic potential, in that cases here we require sodium ions. Okay, so there is a movement of sodium ions from inside of the cell to from outside of the cell to inside of the cell. Okay, so and nothing but influx means there is a movement of ions or substance from outside of the cell to inside of the cell. Then efflux of potassium, okay, means there is a movement of ions from inside of the cell to outside of the cell. Okay, so in case of excitatory neurotransmitter is binds with postsynaptic neuron, in that cases that neurotransmitter will increases the permeability nature of the cations. So that cations is allow the it increases the influx of sodium ions and efflux of potassium ions. So it may lead to depolarization, also known as EPSP, is nothing but excitatory postsynaptic potential. So as a result of this EPSP, then what will happen? Excitatory like effect will be occurs, like muscle contraction, okay, gland secretion, like that. Excitatory effect will be occurs. Next, in case of that presynaptic neuron is releases the inhibitory neurotransmitter. Okay, in case of it releases the inhibitory neurotransmitter. Then what will happen? That inhibitory neurotransmitter, okay, so that inhibitory neurotransmitter will binds with receptors. The receptor is present on postsynaptic neuron. Then what will happen? It may lead to hyperpolarization. Why? Because it allows the permeability of anions. Okay, in excitatory neurotransmitter, it is allows the permeability of cations, but in hyperpolarization, uh, it in inhibitory neurotransmitter, after binding with the receptors, the receptor is present on postsynaptic neuron. This one is inhibitory neurotransmitter, is binds with postsynaptic neuron. Okay, this one is receptor. Okay, after binding with the receptor, then what will happen? Permeability nature of the anions is increases. Okay, so here high influx of chloride ions. Okay, which ion? High influx of chloride ions. Okay, so then the negative charge inside the cell is increases. Okay, and efflux of potassium ions, means slowly the positive charge within the cell is decreases, the negative charge within the cell is increases. It may lead to what? Hyperpolarization. Then we can get inhibitory like effect, like muscle relaxation, glands decrease in the glandular secretion. Okay, like that. Okay, so this one is the transmitter action on post-junctional membrane, okay, or postsynaptic membrane, and postsynaptic activity. Okay, depending upon the release of the neurotransmitter. After release of neurotransmitter, how neurotransmitter is released? After arrival of action potential. Okay, after arrival of action potential, these are the steps it will be happens. So how action potential it will be produced? After when the resting neuron is exposed to stimulus. Okay, when the resting neuron is exposed to, okay, in resting condition, the membrane potential is minus 70 millivolts. But when it is exposed to stimulus, okay, there is a depolarization thing is happen. There is high influx of sodium, the positive charges increases, inactivation of potassium pump, there is a production of the action potential. Okay, so after action potential, okay, action potential production requires stimulus. Okay, so this action potential, then what will happen? This action potential will reach from presynaptic neuron to postsynaptic neuron with the help of what substance? That substance is known as neurohumoral substance, nothing but neurotransmitter. Okay, so the propagation of nerve impulses requires one chemical substance. That substance is known as neurotransmitter. The process of transmission is known as neurohumoral transmission. Okay, got my point? Okay, so this one is the process of production of action potential. Then this one is the process of release of transmitter. Okay, when it is released after arrival of the action potential. Then this action potential is propagated from one neuron to another neuron with the help of this neurotransmitters. Then after releasing of neurotransmitter, so there is a somewhat excitatory effect will be happens in postsynaptic neuron, or inhibitory like effect it will be happen in postsynaptic neuron. So what kind of that thing is depending upon the type of neurotransmitter. That if excitatory neurotransmitter is releases from the presynaptic neuron, means it binds with postsynaptic neuron and it shows the depolarization like effect, nothing but excitatory postsynaptic potential. So it shows the excitatory like effect. So because of the high influx of sodium ions. In case of inhibitory neurotransmitter will bind to the postsynaptic neuron, then what will happen? There is a high influx of chloride ions. So the negative charge within the cell is increases. So it may lead to what? Inhibitory postsynaptic potential. Okay, either excitatory postsynaptic potential thing is happening in postsynaptic neuron, or inhibitory postsynaptic potential thing is happening in postsynaptic neuron. So depending upon the binding of neurotransmitters with postsynaptic membrane. Okay, so this one is the third and fourth step. Next, last step is termination of transmitter action. Okay, termination of transmitter action. Then here it is, synaptic transmission is ends. Okay, when dissociation of the neurotransmitter from the postsynaptic membrane channel. Okay, so the postsynaptic membrane consists of some ion gated channels is there. Okay, so these are the channels. Okay, this one is channel, ion gated channel. Okay, that this one is receptor. The receptors consist of ion gated channels. Okay, so in first step, the termination of transmitter action. So first one is the synaptic transmission is ends. When it is ends, there is a dissociation of neurotransmitters from the ion gated channel. Okay, that time only the synaptic transmission it will be end. And then most of the neurotransmitter is pumped back into this presynaptic neuron. Okay, see here, uh, most of the neurons, most of the neurotransmitters. Okay, so most of the neurotransmitters is inactivated due to it is transported back into the, okay, transported back into the presynaptic neuron. Okay, so that process is known as reuptake. Okay, so third step is the neurotransmitters are broken down by enzymes. Okay, so with the help of some specific enzymes, okay, that will discuss in further chapters like cholinergic system and adrenergic system. Okay, so with the help of enzymes, so there is a neurotransmitters, it will be broken down. That process is known as degradation. Next, the breakdown products. Okay, in degradation process we will get some breakdown products. Then again that breakdown products will enter into the presynaptic neuron. Okay, what is the purpose of entering of presynaptic neuron? It is that breakdown products, it will required for resynthesis of neurotransmitters in presynaptic neuron. Okay, so that breakdown product, that degraded product is also required for the synthesis of again fresh neurotransmitters in presynaptic neuron. Okay, so this one is last step, uh, termination of transmitter action. Okay, so this one is the total process of neurohumoral transmission. What is neurohumoral transmission? The process of transmission of nerve impulses, okay, from one neuron to another neuron or neuron to effector. Okay, so with the help of neurohumoral substance that is known as neurotransmitter. Neurohumoral substance is nothing but neurotransmitter. Overall, that process is known as neurohumoral transmission. Okay, so in neurohumoral transmission, the first one is impulse conduction, means how that impulses it will be conducted from one neuron to another neuron. First thing, how action potential production will be produced. Okay, when stimulus is exposed to that resting neuron, then there is a production of action potential. So then after action potential, after depolarization, then repolarization is happened, then hyperpolarization, the resting state, then transmitter release. Okay, the transmitter release, next transmitter action on post junctional membrane. So then in case of excitatory neurotransmitter is in case of excitatory neurotransmitter is binds with presynaptic neuron, means postsynaptic neuron, means there is a EPSP thing is happen, excitatory postsynaptic potential. In case of inhibitory neurotransmitter is binds with the postsynaptic membrane, means there is a IPSP is happen, that nothing but inhibitory postsynaptic potential, it may lead to inhibitory effect. Okay, then termination. So the termination consists of here 5 steps. The synaptic transmission ends when the dissociation of neurotransmitter from the ion gated channels. Okay, and there is a reuptake process with the help of reuptake process and degradation process, and again the breakdown product will enter into presynaptic neurons. Okay, that breakdown products is also again resynthesized in presynaptic neuron. Overall, that process is known as neurohumoral transmission. Okay, simple. Neurohumoral transmission means the process of transmission of nerve impulses from one neuron to another neuron or neuron to effector organs with the help of neurohumoral substance. Okay, nothing but neurotransmitters. Okay, so Thank you. Okay, so in next class we will discuss cholinergic drugs. Okay, so cholinergic system and adrenergic system. Okay, the cholinergic system is also known as parasympathetic. Okay, and adrenergic system is nothing but sympathetic. Okay, so next class we will discuss about cholinergic neurotransmission. This is a overall neurohumoral transmission example. Next class we will discuss cholinergic neurotransmission. Okay, how acetylcholine is synthesized in presynaptic neuron, and how it is released from presynaptic neuron to synaptic cleft. Then how acetylcholine is binds with the postsynaptic neuron, and what is the reuptake process. The reuptake process requires some specialized transporters is also required, and what is the names of the transporters. Okay, then either it is it is broken down by some enzymes. Okay, what is the names of that enzymes in cholinergic system, in adrenergic system, that we will discuss in further chapters. So in next chapter, in next class, I will discuss cholinergic drugs. Okay, thank you.